long-distance pathways in half range.
Hi. I want to thank the organizers for inviting me to this talk, to this workshop. My talk is mostly going to focus on neuroinformatics. So basically what I'm going to talk about is our work on mining collision of connectivity database. This was work in Germany, with a database in Germany where they collated connectivity information, case study information from a bunch of papers and made it available online for everybody. So that's going to be the first part of my talk. I'm going to talk about the analysis of networks, the network that we found after we mined the data. A lot of the work that we do in IBM is unlike the beautiful work that was presented earlier today and yesterday, is on high-level models. We do not do biologically very detailed models or very realistic models. We are more into phenomenological models. And the reason that IBM is basically interested in this, to sort of give a background, is it started by the fact that we have this large computer, the Blue Gene, and we were interested in finding out what all can we do with it. What kind of simulation that could run, what were the drawbacks of the system, what were the changes that you needed to in some sense mimic or model the brain, not really get down to cognition, but that was the first part of what our efforts were. And there, I think the efforts mainly were on, as I said, on point models or phenomenological models rather, and on large scale, where the testing of how well Blue Gene or a supercomputer did as the scale increased, what was of interest. Over the years, cognitive computing has from the large scale simulations we've evolved. There have been in about 4 years back, DARPA, the US agency, got interested, and primarily got interested in it because the belief that the brain is a very efficient, space and power efficient device. Right? So they got interested in finding out what were the key elements that you could derive from whatever you knew right now as a fact. What are the key elements that you could derive so that you could maybe fabricate those chips, maybe design and fabricate power and space efficient chips, which could do some cognitive tasks, do some tasks which are more suitable for non-one-on-one computing. So I'm going to talk a little bit about that. That's going to be the last part of my work. So to start, so there have been multiple views of the brain, and I'm sure most of you are familiar with this. I think the chronological order is more what is in the middle. You have those sort of local connectivity networks which came out, which were mapped in 2004. 2007, these protein fluorescent proteins to sort of which could map the connection between neurons. Sebastian and Denk also came out with, and they have worked on connectomes for the past few years. In fact, Sebastian has a book on it that came out last year. And you had the DTI networks, which have become popular in the last, again in the last couple of years. So you have the DTI, original DTI data, and then some kind of model DTI data where you have the areas and other things. So this has been sort of and before this has been the tracer studies, right? Which I believe was started around in the 1900s or early 1900s. So there's been a lot of different views of the brain, but one, like in some sense, or at least one largest collection which has been available publicly for everybody to use has been CoCoMac, which was basically based on tracer studies. We'll talk a little bit more about it in a second. So, CoCoMac was basically started by Cotter and basically the citation is Cotter and class in 2000. It's 430 different literature reports. Basically, in these 413 literature reports, 7,000 odd brain sites have been injected with tracers. I'll talk about what are mapping details. So there have been 2,508 tracer injections, different tracer injections, I guess. I don't know if I'm sort of confused about the two numbers, but and there have been about 40,000 odd connection details. Now, CoCoMac basically, from what is basically does this, did this, that they looked at each literature study and they looked at what were the brain sites the tracer was injected into, whether it flowed to so target sites, source site, target site, and then between two different literature reports, they also found a mapping. So they studied, if one literature study was studying some region here, the other literature study was studying another brain site. They found they claimed a mapping between those two brain sites. So let me give you a detail about it. Let me give you an example. So, for example, the first thing is that the data is the brain sites are identified by the authors, the year and the brain region. So, for example, the V1 area study by Feldman and Van Essen in 1991 will be labeled FP91 V1, while the area 70 study by Broadman in 1909 will be labeled by B0970, and so on and so forth. So every brain site that has been studied in these 430 literature reports has been labeled according to the author who studied it, the year that was studied, and some whatever nomenclature they used to describe the site, right? So if Broadman used area 70, they used area 70, and then Feldman and Van Essen used V1, they used V1. Now, obviously, the first question here, okay, let's just talk about what was there. The second thing, the thing that CoCoMac did was they developed a mapping relationship, right? So there are three types of mapping relationship. One was identical, four types: super area, sub area, and overlapping. So each brain area, so they give an example. Each brain area, a brain area like Feldman Van Essen's V1 was considered to be identical to Broadman's area 70. So this was a mapping relationship that CoCoMac decided, and similarly, they decided that Feldman's V1 is a sub-area of the occipital lobe. So they went in, some of this is based on the literature report itself, the description of the literature report, maybe the relationship that were reported in the literature report. Some of it is based on their own judgment. But this is how the mapping relationships came about. The reason why you need a mapping relationship will become clear in the next slide. The third database that they had was what is called the connectivity, the actual tracing study database, right? So, and the connectivity database was basically the source brain site where the tracer was injected, the target brain site where the tracer came from, sort of flowed to, and some kind of a notion of strength, some kind of lamina information, but that there was a little bit more, but we are sort of ignoring that now. So, for example, they would say that there's a tracer study which says in the paper, I don't know who the authors are, in 2002, there's a paper which reported a projection between V1 and V2. And there's another paper which is basically reporting a connection between area 1 and area 46 in the front lobe. Now, these are the three main data sets that we extracted from CoCoMac. Before I go further, basically, let's just, I want to sort of, the one way to think about this database, entire database, is to think about it as a mapping of the world kind of a thing. So you have brain sites, which you could think of them as countries, and you could have those at country level, at the state level, at the city level, or continent level, or at any level, depending on the resolution of where you're drawing the line. Then you have basically different people who are drawn an atlas, right? So you have an atlas which is drawn in 1909, we have an atlas which is drawn in 2002. Maybe some people drew an atlas only of Australia, some people drew only an atlas of North America. So there are people who have studied different parts of the brain or different parts of the atlas. So that's and CoCoMac's contribution is they basically looked at all these different atlases and sort of said there is a mapping relationship between the two. And the third thing is the actual connectivity. So somebody has gone into the atlas, using the atlas, they have said, okay, there exists a trail route or a plane route or a highway between these two brain sites. So now you have this separate atlases, each of them sort of having a different nomenclature. You want to put them together, and then you want to, all these routes that you have, whether they're highway, train, plane, you want to sort of make sure that they kind of line up and become consistent with the atlas that you're drawing. So, so I'll give you a better example of that in this slide. And basically, what this at the top in CoCoMac, there would be a connectivity tracer study mapping thing. Then you would see a disjointed. You would see that okay, there is a forward projection here, there's a backward projection here, but you wouldn't know that they are basically reciprocal if you didn't consider the mapping detail. Another example of the same thing is that basically CoCoMac. So when you see the arrow, that's what CoCoMac is reporting. So this part CoCoMac reports, this part CoCoMac reports, this part CoCoMac reports, and this part. So all these arrows report. But when you make the bounding box, which is the mapping details, you are basically able to figure out that these are identical, these are identical, these are identical, and then you are able to find a very nice cycle. Basically, V1 with visual areas to I'm not a neuroscience, so excuse me. TF and then hippocampus, I guess, and TH and then back into visual area. So unless you are able to do the mapping relationship, unless you are able to sort of put the atlases together, you are not being able to merge the paths and make the right paths, be able to find out where the paths lie. And this whole thing is because a lot of people have studied at a lot of different resolutions and at a lot of different and in a lot of different ways, and use a nomenclature which was maybe what they were, maybe their community was studying, or but a lot of different nomenclature has developed over the years in neuroscience. Now, we started this work in 2008, and CoCoMac has been around for about 6 or 7 years before we were this thing. And one of the reasons why not that much work has been done in mining CoCoMac was because basically, because of these mapping relationships, there were conflicts. So I'm going to now talk about what the conflicts were and what basically our contribution in mining the CoCoMac database came about. So the contributions, the conflicts could be very simple. That in the database itself, there could be one study which could be saying that PFCD is a subset of area 9. And there could be another study saying PFCD is equivalent to 9. Right? So basically, let me just, I'm going to concentrate only on 2 mapping relationships. One is identical and one is sub-area. Right? So if you think of it as a parcellation, all you're concerned about is those which are identical to each other and what is sub-area. So CoCoMac would give you, by itself, there could be some, this could be because of human error, but this could basically be because different literature people had thought about it in a different way. Right? The other thing is, so identical, both identity and sub-area are transitive relationship, right? A is identical to B, B is identical to C, so supposedly you should say A is identical to C, right? Now, so that transitory also will create a problem. So if somebody says PCI is identical to IPL, is identical to PF-1, is identical to 7P, but there's another literature report is basically which is saying that PSCI is a subset of 7P, right? So that's kind of a conflict right there. The conflict that arises here is transitivity, right? So these are simple conflicts to deal with, and there are more complex conflicts to deal with right here. Transitivity conflicts. Similarly, there could be a bunch of them. Even out here, basically, you can't, I don't know how well you see it. Transitive T will give you these blue spots, and everything that is underlined will give you a closure. So, will give you a conflict. So basically, when we were mining this database, we found that there was some simple conflict, but a lot of transitive conflict. And Stefan and Cotter knew about this because when they published the original paper, they had talked about it. It's not something that we came up with. by the very nature of establishing it, it is difficult to find out transitive complex, like when you're setting up with database, right? Especially the way when they're looking at different literature reports, it's not that they can sort of look ahead so much and say, okay, this relationship might occur later. So it is a difficult task, and for both from this way of putting in the data and for cleaning it up, whichever way you look at it. So basically, the biggest contribution that we had is in mining the database, and the biggest step that we came up with was something which was based on what is basically called transitive closures. If you take a set, especially if it has a transitive relationship, you basically find a closure of it. Closure meaning there's nothing outside. If you're saying A, B, C, the example that I gave, A is equivalent to B, B is equivalent to C, then A and all three will belong to the set. That will be the transitive closure, right? So there's nothing outside of the set, which cannot be reached by a transitive relationship within the set, right? Sorry. So the idea was very simple. So think of I as the original set of identical relationships, L as the original set of subset relationship. You find the transitive closure of I, you find the transitive closure of L, right? So L plus and I plus are the transitive closures. And the transitive closures are very simple to do if you have a matrix. It's basically parse the matrix, removing the diagonals every time that you take parse the matrix. The conflict that you're looking at basically the intersection of I plus and L plus. Basically, wherever you're coming up with this is the ones that are all the conflicts that are there in the data set, right? And these conflicts exist, and so what? Now the question becomes, what is more important for you, I or L? One of the judgment calls. So in our case, we basically said I is the more important relationship. So we started with a set J, and we said, let's start this. I haven't drawn it very well, but let's just start with a small set J and keep sort of increasing it till whatever maximum we can increase, so that J plus never intersects L plus. Right? So then you can't have conflicts. You are trying to find a big set of identical relationships which do not lead to conflicts. This was the main insight, the main this thing that we came up with when we were doing the mining, and this was the reason that we were able to get a lot of data out from CoCoMac as compared to what had been taken out previously. So what did it lead to basically? What it led to, I'm going to give you, this is the first of the parcellation scheme or the map or the atlas so to speak. So we came up with one map instead of so many different atlases. There was one map. So where the resolution, the highest resolution, it was a hierarchical map. There's a brain, there's a cortex, which is divided into these lobes. All these acronyms that you see are basically consistent with whatever they're in CoCoMac. So all our acronyms are, in that sense, we can go back, take if you look at the paper, you can basically look at, so for example, you can query CoCoMac and CG #2 and you can find out all the information. So that way it is consistent with whatever is in CoCoMac. So brain is cortex, basal ganglia, and diacephalon. Cortex is divided into these different lobes. Example of further division is occipital lobe is divided into V2, V1, and then back and OA. And so this is basically based on the J plus and N plus, whatever relationships that we could come up with, right? So each of these areas, from the 7,000, each of these areas would have other areas which are identical to it merged into it. Right? So if V1 is identical to 17, all 17 connect, if V1 would be most probably identical to 17 and some other areas, so all connections which are reported for 17 or other areas will be merged into V1. So each of these areas had other areas which are equivalent to it merged into it. And then they were sort of established in this hierarchy using the L relationship. This was the temporal lobe. Basically, again, the hierarchy arrangement, the thalamus. And this was the entire hierarchy, right? So we came up with this radial tree layout. And you have the brain, the cortex, you have the lobes, you have the further subdivisions. Now, this is a question, actually I'll come to that question a little later. So you have the good thing about it is not, you see that it's the entire brain, you have the thalamus, you have the frontal lobe, you have the basic ganglia. It's not just the cortex, the other parts of the brain also in this. And there's one parcellation. Instead of having multiple parcellation, multiple disjoint parcellation, or overlapping parcellation, there's just one parcellation. It could be argued that this might not be the best parcellation. There could be other different parcellations. So be it. There could be, but this is the way this is the one that we came up was most consistent with the methodology that we use. Now, till now, I'm talking about the atlas of the parcellation. Then, obviously, the most important thing is not the parcellation, the actual connectivity, the projection. Right? So, on this parcellation, basically when you draw one projection, I guess it's the projection between V1 and LGN. That's one. So basically, between each node in this parcellation, there could be a connection. Right? Obviously, the nodes which are internal should not have connections, but unfortunately, there will be nodes which will have partial connections because somebody might have studied an area at a high resolution. Somebody later on might have studied that same area at a much lower resolution. We have not decided to remove those connections because of lack of knowledge. So we have reported all connections. So there are this is, if you draw a 9-bit representing a connection, so this is one connection. There are 6,600 connections. So it's pretty, visually it's a dense graph, but you could do bundling and you could draw it in a smart way and you could come up with this nice diagram, which is basically all the 6,600 connections kind of bundled together in different ways. This is not the same bundling used in the brain, or this is just visual bundling. Based on the hierarchy, you kind of use that as for splines to bundle the data. How does this whole thing compare with whatever was available? So whatever was available was, as far as we knew, publicly available was these data sets at the time that we came up with, and basically ours was a jump, substantial jump ahead, right? So we have many more areas and many more connections between areas. And to put it visually, basically, Kaizen and Hilgetag, who had mined CoCoMac also, this was their data. Basically, one of the things is that they were not studying, they were studying in the cortex. They were not studying other. But this was the other best known publicly available data set at that point of time as compared to this one, which is definitely fuller. Now, one of the things we sort of kind of we stress upon this point. We are comprehensive in that we have not started any study in CoCoMac. So our background is computer science. Both the papers, both the people who wrote this paper, we had no knowledge of neuroscience, so we had no intentions of making any call on what should be included and what should not be included. So whatever was there in CoCoMac was included in the data. You can trace back every connection. So if you look at the paper, there is a very clear where there's all the data is given in a way that you could sort of query the connection and take it back to CoCoMac and see where it came from, which radiation reports is supported. It is concise because we started with I think this is the 7,000 odd brain sites which were merged together, which were identical to 300 odd brain sites, right? And there's one parcellation of one atlas, and colossal is just our selling point. We like to say that it's large, but so based on so this was the mining part of it. The mining part, the chief algorithm was the transitive closure and how to basically how to ensure that you could use transitive closure to avoid conflicts. The second part, now that you had the network, this network, so basically you had 383 sites and you had a connection with some of those sites, 6,600 connections. So you had a network, right, an edge. This is just the presence of a projection. It does not talk about strength, it does not talk about the laminar, many other things. It's a very high level. Just say that some study in CoCoMac, there was a study where there was a projection from area A to projection from area B. So we basically analyze that network, and these are favorite network theoretic methods to analyze this thing. So two of them start basically the diameter and the characteristic path. Well, three are basically related to what is called the small world model, right? So basically, the fact is that you could have a large, long diameter, which is the shortest path length, but then you could know you could have the two things that you require is a high clustering coefficient and reasonably high diameter. That's the small one effect, right? So you're getting as compared to a random network. The other thing that is there is that the question, this is a directed network where there's a direction between the source side and the target side where the tracer flows from. 42% of the connections are reported to be this thing. Again, there could be issues about whether there is, what kind of directionality is there, whether some areas have not been studied enough, whether certain projections have not been studied enough, but this is what the data reports. This is a famous, Warren Beaver was a philosopher, scientist, and I guess he was also, he dabbled with a lot more in computer science. And he came up with this concept of organized complexity of modules, of modular systems. And basically, the organized complexity is a very simple model where it says, take a system, if you can divide it up into modules, it's great, right? Because, breaking up things is great. But now if the modules are very well interacting a lot among themselves, then what does it mean? So his whole point was that when you look at when you organize them in this kind of a matrix form, is the off-diagonal element, which basically represents the intra-module interaction, is also high. What does it mean? That's the concept of organized complexity. And he came up with this term of organized complexity. This organized complexity is good because these modular systems can be then simulated well using computer systems. That was the point of Weaver. So here you find that if you look at all that off-diagonal elements, most of them are pretty well. There's a lot of connection with acid from the frontal lobe, temporal lobe to basal ganglia. I guess that's also frontal lobe to temporal lobe. So, though they are modular systems in the sense that you can say there's a temporal lobe, there's a frontal lobe, but each of them are interacting a lot with the other lobes also. And the other results, which say network theory, the other big result that has come out. Basic network theory has two major results in the past 20-25 years, I guess. 20 years, small world effect and scale-free graphs. Scale-free graphs are very popular because everybody says if you draw the degree distribution, the degree is the number of edges that come up per area, and you draw it in a log scale, everything will be a straight line. And scale-free, Barabasi made them popular by saying that scale-free networks come about because of preferential attachments, right? So the preferential attachment for anybody, for everybody who's on Facebook and all know that basically there's no comment. If you're popular, you can get more and more edges or more and more friends very easily, right? So in social network that makes a lot of sense that you can get more and more popular. It's also called the Madonna effect. It's called the various names for it, right? So preferential attachment leads to scale-free graphs where the degree distribution falls in a certain way. Unfortunately, and it has been hypothesized the brain was scale-free. Right? People wanted to say that it was scale-free. C. elegans has been shown to be non-scale-free, but they were hoping the primate brain was scale-free. There was no data on it. So when we drew our degree distribution, it came out to be non-scale-free. It wasn't a perfect line in the thing. On the other hand, when you do it at the semi-log scale, it goes to straight lines. So it's exponential degree distribution, which makes a lot of sense if you think about it. Say scale-free or preferential attachment is very easy in a Facebook kind of environment because There's no cost of adding a friend. You can have as many friends as you want. And the moment you have a cost where you cannot sort of add friends or add edges or add add friends in that or adjacent neighbors, you will not have scale figures. And you would have exponential where you will basically have a tail. So this is more consistent with the fact that in the brain you can't sort of keep adding it and keep adding more and more connectivity. So this was one result that we came up with, we saw, which is also consistent with the CLM's result which came out many years ago. Another example of network mining, which it's basically that what happens is that people look at how nodes are important. Right? So what is the concept of node being important? It could be very simply degree. How many edges do I have? How many friends do I have? Or it could be as complicated, and I think this is the example I'd like to take is page rank. Page rank, I think some of you will. Page rank is the algorithm which Google uses to give you search results. And basically the way page rank works is that a page is important if it is linked by important pages. It's just not important because there are lots of people linked to it, right? Other important people, so it's kind of an iterative definition. You're as important as your friends in a social network kind of scenario. So, and between us is a concept, the shortest path. These are all very similar concepts in the sense they all look at network, look at the paths, the hop distances, or if they are weights given, they basically look at some of this thing and they try to find out what are the important paths, what are the important edges in the network. And what we want to say, what I want to say about this graph is basically about this table is that when you do a very simple thing, again, this is just basically saying there are 8 ways of measuring importance of a node. If you put them and you rank the top 10 for each metric, then in this table, which is 10 into 80 entries, most of them turn out to be from prefrontal cortex. So there's nothing. You just take the structural data, just do this simple analysis, and just do the top 10. Quite a few turn out to be. The majority turn out to be prefrontal cortex. So from our knowledge, prefrontal cortex is supposed to be sort of central to a lot of tasks in the brain, and structurally also it is really important. That sort of now continuing along the line, basically we continue to ask what are the topological importance, what is the way to measure, what is really just in a topological analysis of that network. So one of the ideas is that supposing you take a local connectivity. So a lot of people even the previous results people used to talk about important nodes and important what is central. Now if you forget that and you just say I want to know what is around me, it doesn't make a difference if I'm important to others or not. I'm just concerned about myself. What role am I playing really? Am I sort of connecting to people? Am I working by myself? Am I doing nothing? Am I just this thing? And here the concept of what we call stars comes about. So if you take a local network and if none of their adjacent neighbors are connected, then it's a star, right? Because they all, like, think of it as you're the center and there are 5 or 6 adjacent nodes. So in that case, if that's true, then you are playing an important role in sort of providing connectivity, communication between, enabling communication between these people. If that's, on the other hand, if all the adjacent people are talking among themselves, then you're not providing any connectivity, and that's a clique, right? In a clique, it's not, communication is easy. Anybody can talk to anybody. There's no real one person who's enabling it, right? Clique is, in some sense, clique is about computation, star is about communication, right? Clique is a different. So here what we are drawing is basically, it's very simple. For each vertex, we are basically saying, just tell me how close am I to a star? Am I doing a lot of work in terms of communication? Am I enabling a lot of communication between people? If you're at this part, then you're doing no work. It's sort of inverted. This thing, if you're doing out here, you're doing a lot of work because a lot of people, you have a large star and you're sort of enabling people will talk to them. So this basically the profile, the reason which we find very interesting about it is, it's kind of nearly continuous. So the brain really doesn't sort of optimize a local network. It doesn't seem to. Every local network seems to be is distinct as compared to when you look at constructed network. One best example being an internet network, where there's a preference for a certain type of network. They kind of, and the reason that there is preference for a certain type of network, because in a constructed network like this, constructed and not overly optimized network like internet, basically a copy model is often used. You basically say, okay, this is the best router system out there. They have done this kind of service for a while. Just let's copy the same router system here and copy its neighbor, and it will work well for our systems also. So that kind of thing, if you see a lot, and there are other networks that you see, constructed networks that you see, this kind of staircase structure. While in the brain you see a more regular profile. The last thing is that in the brain there are no pure stars. There are cliques in the brain, but there are no pure stars in the sense that there is no one neighborhood where somebody is playing the role of sort of enabling communication between others. So our conjecture is that basically, it is true that the brain optimizes for violence, but it also may be true that the brain is working and providing connectivity. It doesn't just optimize on violence. It enables connectivity when needed. This is I'm going to go quickly through this one. This is very recent work that we are doing now. This is the same graph that you saw earlier. The splines are drawn this way because basically these are the intra-lobe and the intra-basal ganglia or the intra-thalamus splines. So you see in the thalamus there are really not that many intra-connections, while in the frontal lobe there are a lot of connections. Same way over the temporal lobe. Now the thing is that on top of it, basically we are saying that there is a concept. Again, we are looking at local connectivity. What am I enabling? And in that sense, what you could have different kind of roles. This has been done way back in social networks. You could be a gatekeeper, where you could be a coordinator. Basically, you're allowing two of your fellow group members to talk. That could happen. You could be a representative, where all the communication is happening from your group is happening through you. You could be a gatekeeper, where all the communication to your group is happening through you. And you could be a consultant, which is between two different groups. And you could be and the consultant is between, you will be a different group, and the other, you will sort of act as an external guy to another group, and you could be liaison where it will be two different groups. Groups here are basically the groups that we have decided is the groups of thalamus, basal ganglia, the lobes. So there are 8 groups in this network. Just a very simple kind of this thing. And based on that, we have finding this thing. We are still to sort of analyze, but we see out here in our occipital lobe a lot of well-defined, occipital lobe seems to be very balanced in the kind of structure, roles that it does, while frontal lobe is a lot more coordinated and stuff. This is something that we are digging deeper and working on it. So this basically kind of includes what I want. Well, this is a kind of network theoretic or network work that we are doing. Basically, taking the topological network that we mined from CoCoMac, and we are sort of trying to make different, based on the data, topological analysis. Like what are important nodes? What are the roles that nodes play? What are the roles that they have communication? What are the different? What are they really trying to do based on this topology? Obviously, there's a lot more neuroscience data, right? There's layers, there's all those things which we don't have the data for. So if and as we get that data, we can do more about it. I'm going to spend the last few minutes talking about this. What really sort of pays for all this, what I interest in this. So the reason why IBM was interested in this is basically what they call the cognitive computing thing. This, as I said right in front, this is sponsored by DARPA, and there's been a project for the past 4 years. I'm sure all of you are familiar with approximately these kind of other things. Basically, by processors we mean the neurons, the interconnection of the synapses, and the wiring lens the axonal length. Obviously, there is this amazing capability to do this, and this question of whether we can have computers who can build this, can do this, is a question that we are not the first to ask. And I'm sure we'll not be the last to ask. I'm sure there will be more work on this as we go along. What fascinates IBM in general, or fascinates me in particular also, a lot is the fact, basically captured by this thing. Basically, you have all these sensors, right? And there's a lot of data coming, and I'm told there are neurons in your feet, there are neurons in your hand, there are neurons you're seeing something. There's so many people out here, you're sensing so many different colors, faces, and as you're standing, you're sensing, you're self-balancing it. You're hearing things, there's something happening out there. But still, you're able to still do action. So there's so much different data coming in, right? The best computer that is out there right now cannot basically process this amount of, forget what it does with it, forget the fact that it is cognitive or intelligent, or it cannot just load that kind of data that is coming in. When we talk about this, I'm sure, given the pervasiveness, all of you have seen the Smart Planet advertisement that IBM runs, right? And think about if you are going to throw out sensors every corner of the world, and that they are going to send out that kind of data. Can you really get that data, and can you really do maybe say simple linear regression? No, you can't, because that volume of data, the throughput of data is too high. But it seems the brain is able to do. Whether through attention, whether through some selection, whether through whatever mechanism, it is able to process a reasonably large amount of data, right? And it is able to process it in a power which is less than a light bulb, and space space which is less than a liter bottle. So this is important because of the fact that I don't know, I forget the number, but recently only about last week there was a number about how much power consumption data centers are taking. All of us use Google. Every query that you do through Google costs a certain amount of power, right? Each data center, nobody releases the number, but somebody estimated the number. Right now, the data centers are the largest consumers of power in the world, outstripping all other. The magnitude is higher than any other industry right now. It's not aluminum. Everybody used to say aluminum. Aluminum. I forget what it's called. Smelting. That used to be the number one. But data centers as this thing have become the number one by a large margin. So obviously, for all companies, doing power is efficient. Space is efficient more because of the fact that if you put brains on a chip kind of a thing, or do something like that. This is how it came. Basically, the idea was that when they looked at the current devices available and the current technology available, they said that the numbers, just in pure numbers, again, and again, I have to emphasize that this is the thing. The pure number and phenomenological models, not detailed models, not those kind of things, could be implemented in silico. Just based on these numbers, and this is a reason the project in some sense. So, what the state of the project is that there is what is called a cognitive computing chip. It has 256 neurons, and so 256 into 256 synapses. It has programmable synapses. Currently, it basically plays Pong. It doesn't do much more than that, but it has the ability. There's work going on currently on vision. There's work going on navigation and other kind of things. I think that's about it. And the reason why we kind of relating it back to CoCoMac is why we studied all this was basically the idea was that we take these chips, each area will become a chip, and we sort of put them together and then see what really comes up. We haven't reached that stage yet. We're hoping to do that soon. How will this be used? in a typical scenario is that you think of this as a smart bandit kind of thing. Some data will be coming in from here. There'll be contextual data that you had, and basically there'll be feedback, and so there's sensation that's coming in, and you can have actions. There's the various examples of this. One is the healthcare, where basically you have a lot of different kind of time series data coming, right? So you have, there are two places where, one which will make you a lot, both which will make you a lot of, one is the financial data scenario where there is a lot of time series data coming in, and the other is healthcare. And obviously, the healthcare kind of a thing, it helps to have this brain on a chip kind of thing because it's small. Right? So that kind of concludes my talk, I think, yes. I'll just close it with, this is a quote that actually was in a magazine, but it kind of has captured our philosophy in cognitive computing, so we like to put it at the end. And I'll ask the questions.
This is, of course, extremely interesting that you have been able to develop this for CoCoMac and the that comes out. Of course, as neuroscientists, we all wish to have a Google macaque and a Google mouse and a Google worm perhaps. And but to have that, you need to federate this to different other databases, like the connectivity and the columns and the sort of the microcosmos of each column. And do you foresee a development in that direction within your framework? Because I think that probably very critical information also for the second part of the talk.
Yes, I think I see the thing. To put it, currently a lot of IBMs, a lot of our efforts are being spent in sort of mining the data. In some sense, we are not giving back. We are publishing the data that we mine is publicly available. But what you're saying is right. So, for example, when you simulate, you need this micro-column structure and others. That kind of stuff we haven't been able to really. It is quite complex, and we haven't got a good grasp of it yet. So I think it will become maybe say this project is supposed to last another 5 years or so. So in that timeline of 5 to 10 years, we do expect that we will be able to sort of provide more rich information based on whatever our observation or whatever our experiments were. So this will be more like I think the way it is being right now is this is. We took some neuroscience data and we did some simulations of mined that data, and now what we give back is to neuroscience, does this make sense kind of a thing, and they can then say validate it and come up with maybe better models, or maybe they can study it in more detail. But it hasn't started happening right now.
So IBM was involved or is involved with the Blue Brain project in
Right.
Is that completely unrelated to your effort?
So Blue Brain is a very good example of detailed models, right? That they are really going into details. Right now, IBM is not involved directly, as far as I know. But they're doing a great job of sort of going right into the neuron and the other structures, synapses and neurotransmitters and all. So I think it's sort of their part. The way we think about it is the microscope and we are sort of a telescope. I don't think they are deep and we are sort of standing out and watching them.
But they should be noticed.
So the cliques that you're talking about, are they related anywhere to dynamic code, calling
So the Trinonian elements were we also found a code. So there's one code in the network which is the tightly integrated, which has cliques, but it has other. It's a code by the sense that if you break it anymore, it will sort of fall disintegrate. there will be no connection.
This is PHC. Is there a current?
Yeah, it has a bunch of areas. So it's that is there. Cliques here, the cliques that I was talking about is basically local cliques. So anything that is within a certain in a local section.
The question is actually a basic one, in the sense that most of the neuroscience computational work involves the hardware of the brain, the spike trains and all kind of things. But we also need software. So we need some data representation in the brain, and now when you make a cognitive computing machine, then you need to know what is the mathematical structure that is running the brain. So has IBM focused on that also?
See, so to be, I think I was having this discussion on what we have, and I think there's a slide somewhere, but what we have, what we've taken from neuroscience is the concept that there are spikes. So you communicate only when you communicate. The processors just don't keep talking to each other. They communicate with spikes on the when there's a need for communication, right? You're taking the circuitry, this CoCoMac circuitry or local circuitry kind of thing. It's very fascinating, you know. This is a circuitry, just to give a very short. People discovered circuitry or the way circuits should be laid out independent of what brain did before, and later on discovered that the brain also does that kind of stuff. So there's something called Rent rule that's there in the brain, that's there in the chips, both there. So there's circuitry, there's communication, and then there's a concept of basically the synapses, the memory or the data being close to the neurons, which is the processor. The data and neurons, data and processing is very close together. Now these are the concepts that have been taken, and this is what IBM's cognitive computer basically. Neuroscience in general, or the thing that you're asking for, has much more to discover and to understand. We are not there yet. We are not talking about. So basically, all we need to know, in some ways, when you talk about software, is how do you translate something into spiking language, into spikes, into process. There is more, but we have not got there yet.
One last question. We are running out. Yeah. From, I guess, point of view of power consumption and so on, are you thinking synchronous?
This is
asynchronous. And scalability of asynchronous circuits and so on, is that an issue at all in your design?
We tested as we go along. Right now at this level that we are, the chips, the one that we have is 256 neurons, 1028 neurons, it's fine. But as we go along.
Okay, let's thank the speaker again.